the previous video, we looked at all of the organs that are involved with the digestive system, such as the alimentary canal and all of the accessory organs. So now we are going to look at the process of digestion in a bit more detail. So digestion is when insoluble food is broken down into a soluble food that can be absorbed by the body. So we want to get the food from a state that it cannot be absorbed by the body to a state that it can be absorbed in. So there are two ways that this digestion can take place. It can either be mechanical or physical, or it can be chemical. So if we look at mechanical or physical digestion, this is when food is broken down into smaller pieces. So this type of digestion then includes the chewing process, also known as mastication. So here the tongue and the teeth break food into smaller particles. Then the bolus formation, so this is when food is mixed with saliva and rolled into a ball before swallowing. So bolus formation, it's very important to know that name. So it is when food is swallowed, that ball shape that it is taken up is called the bolus. So churning movements, the next one, this is when the stomach muscles contract and relax. So food is mixed with enzymes and the gastric juices. A bit later on in the video, we'll look at all of these enzymes that are involved in the different areas of the body with digestion. And then lastly, peristaltic movements, which is when the muscles contract and relax and move the food particles down along the digestive system. So just looking at these two diagrams, so this is a very basic diagram showing a peristaltic movement. This is from the answer series grade 11 life sciences, and it's on page 2.14. So basically, as you swallow the bolus, as the food goes down from the mouth, the muscles behind the bolus, they constrict, and when they do that, they push the bolus down the esophagus. And when this constriction happens at the back, this means that the muscles in front of the bolus need to be relaxed in order for the bolus to be able to move down smoothly. So this process occurs all the way down the esophagus until the food empties into the stomach, which then brings us to the next portion, which is chemical digestion. So this is when large insoluble molecules of food are broken down to a soluble form, very important, by adding water. So water is needed in this process, and this process is a catabolic reaction. So those of you who have physics, physical sciences, you'll still learn, as far as I know, about catabolic. You haven't done that yet, catabolic and anabolic reactions. So catabolic is when something is broken down, and anabolic is when it is built up. So in this case, we are breaking food down, so it's a catabolic reaction, and this is called hydrolysis. And for this reaction to happen, we need enzymes to catalyze the reaction. What is a catalyst, or what is this process we are talking of? So a catalyst can be added to a reaction to increase the reaction rate without getting consumed in the process. So these enzymes just kickstart the whole process, but they are not used up. They don't form part of it. So the enzymes, you need to know some of these enzymes that are involved, and the three major enzymes are for the three major groups that will be processed, in the digestive system. So the first one is carbohydrates, then proteins, and then lipids or fats. So carbohydrates, the enzyme that helps with the digestion of carbohydrates, very easy, carbohydrasis. Okay, so carbohydrasis, and then you also need to know where they are produced, in what digestive juice they are found, and what the end product is of that digestion. So carbohydrates, their most simple form is glucose. That is the end product of digestion there. Amino acids will be the end product of proteins, and then glycerol and fatty acids will be the end product of lipids. So proteins, you'll get to know a bit more about them next year in grade 12 when we do DNA. going to look at the whole process of protein synthesis, so the production of proteins, which is quite interesting. So just getting back to carbohydrates, carbohydrasis is the enzyme that is used here. So it is found in the mouth, specifically the saliva is a digestive juice. This, I can't remember if I mentioned in the previous video, but saliva actually starts digesting, I think about 30% of carbs, I'm speaking under correction, in your mouth already. Then you've got pancreatic amylase. So this is produced in the pancreas, and it is found in pancreatic juice that is secreted into the small intestine. Then you've got maltase, sucrase, lactase, all produced in the small intestine, and they form part of intestinal juice. Next, proteins, proteases, very easy to remember, close to proteins. So you've got pepsin and renin produced in the stomach, and they are part of the gastric juices. And then you've got trypsin, which is produced in the pancreas and forms part of pancreatic juice. Then lipids, lipase is the enzyme, and it is found in the pancreas and in the small intestine, and it forms part of the pancreatic juice and intestinal juice. And then the end product there is glycerol and fatty acids. Remember, when we look at a fat, you've got the glycerol, and then you've got those 3 fatty acids that are attached to that glycerol. So we're just literally breaking them apart with lipase. Then I just got a little diagram here, or a little picture representation. Carbohydrates will become sugars or glucose. That is the name we use. And then fats will become glycerol and fatty acids. See how we've broken up that structure. Then proteins will become amino acids. A chain of amino acids that are more than 50 will generally constitute a protein. Now, let's look at the process of absorption. So we've done digestion. So absorption, now we've digested this food. How do we use it? How does our body get these nutrients and distribute them around so that every part of the body has nutrients to use? So when food has reached a soluble form, it can be absorbed into the bloodstream. This occurs in the small intestine. So this is where absorption occurs, in the small intestine. And the small intestine is specifically adapted for its function in the following ways. So firstly, it is very long. So the food has a long way to travel. Now I put this picture of a 17-seater bus at the bottom because your intestines are about as long as this 17-seater is, and they're all coiled up in your stomach. So it just gives you a nice little reference of how long it really is. So this ensures maximum time for maximum absorption. In the previous video, I used the example of somebody traveling from Witbank to Pretoria on the highway. It's straight and long, and you're going to be traveling very fast. And then you've got, for example, somebody going to Hoedspruit on a mountain pass that's all coiled, and it's also a very long road there. So it just allows maximum time for the food to move slowly through the intestines so that everything can be absorbed that needs to be absorbed. Then next, there's also folds and villi. So the millions of villi and folds found in the intestine enlarge the surface area for absorption. The villi also absorb this soluble food. So as we have the intestine here, on the inside of the intestine, you've got these finger-like projections sticking out. And on these finger-like projections, here's the one on the right, you've also got even smaller projections coming out of them known as microvilli. So this is very important, and we'll look at this structure in further detail just now. Now, how is the villus structurally adapted for its function? So villus is singular, villi is plural. So let's look at how this is structurally adapted. So the columnar epithelium is only one cell layer thick. So this is for easy diffusion, and it also has mitochondria to provide energy for active transport. So if we look at this structure, The columnar epithelium are these little blocks that you can see going all around the villus. So it's only one cell layer thick because certain nutrients will go through this with the process of diffusion, and others will be through the process of absorption, active and passive transport. But more on that a bit later. So, blood capillaries for absorption of amino acids, glucose, vitamins, mineral salts, and water. So the blood capillaries, if we look at this one down here that is in color, so you've got an arteriole, which is a smaller part of an artery coming in, and then it branches into this blood capillary system that is around the center structure, which is the lacteal. So we've got blood capillaries, and specifically amino acids, glucose, vitamins, mineral salts, and water will be absorbed here. Which one are we missing? We are missing fats. Okay, but we'll look at that now. Then lacteal for fat absorption. So because the glycerol and the fatty acids are too big for the blood capillary network, they have to be absorbed by the lacteal, which is part of the lymphatic system. Then next, goblet cells. So goblet cells here on the right, there's one over there. So the goblet cells form mucus secretions, so this helps the nutrients to dissolve in this mucus. Then we also have microvilli that increase the surface area for absorption. They're not very clear on either of these diagrams, but the microvilli are basically all around the villus, and they help for absorption to increase the surface area. Then there are two structures that are not listed here, but the first one is the crypts of Lieberkühn, and then the next is Brunner glands. So the crypts of Lieberkühn basically secrete intestinal juices that will help with the digestion process, and then the Brunner glands secrete an alkaline substance that will neutralize the acidity of the chyme that is coming in from the stomach into the intestines. Because remember, the food has now just been mixed with hydrochloric acid and all of these gastric juices that help with the digestive system to get those foods into a soluble form. So the Brunner glands secrete an alkaline substance just to neutralize the acid in the chyme. And I remember the difference between which one secretes what just by looking at the Brunner glands. That B, I remember the B is for base, and alkaline substances are a base. So I remember that. That's how I can distinguish between the two. Now let's look at the absorption process itself. So absorption happens in the small intestine, like we've said, and in the villus. So glucose, amino acids, water, and other nutrients are actively absorbed against the concentration gradient into the blood capillaries. So they need energy to go across. I explained it in class that pretend you're floating down a stream. If you're going with the stream, you will be not exerting a lot of energy, so you'll be floating passively. Whereas if you're swimming up against the stream, it will be an active transport example, because you will require energy to go against the flow. So glucose is actively absorbed against the concentration gradient straight into those blood capillaries. Then amino acids are also absorbed actively against the concentration gradient into the blood capillaries. Water is absorbed into the capillaries through the process of osmosis, when something goes from a high water potential to a low water potential, and this is also going into the blood capillaries. Then the glycerol and fatty acids, remember I said they are too big for the blood capillaries, so they will go into the system through the process of diffusion, when something goes from a high concentration to a low concentration, and they then go into the lacteal, into the lymphatic system. Let's look at where these nutrients go. So now we're going to look at the transport of these nutrients. So glucose, amino acids, vitamins, mineral salts, and water are absorbed by the capillaries in the villi, and these capillaries eventually become larger veins that form part of the hepatic portal vein. Now the hepatic portal vein takes nutrients to the liver. So eventually this blood capillary network becomes a venule. This venule will then become part of a vein, which is a bit bigger. That is very horrid writing. Become part of the vein, and that vein, so if you look at the right bottom of your screen, you'll see you've got the intestine here, then you've got these little portions, little branches. Those are the venules, and they all come together and they form a larger vein, and then this vein goes to the liver. It does not go to the heart like you learned in grade 10. This specific one from the intestines goes to the liver, and that is why it's called the hepatic portal vein. So hepatic has to do with the liver, and then portal is like an entry point. So the hepatic portal vein, this vein goes into the liver. So let's see what happens in the liver. So in the liver, all of these nutrients that have been absorbed, the glucose is converted into glycogen. So excess glucose is converted into glycogen and stored in that form. Excess amino acids undergo the process of deamination, you need to know that, and form urea and glucose. Now urea is taken to the kidneys and excreted as urine. This we will look at in more detail when we look at the excretory system. The remaining glucose and amino acids are transported to the vena cava, to the heart, and then the rest of the body. So after all of this has taken place in the liver, the remaining glucose and amino acids go out of the liver through the inferior vena cava. So that is now the major vein going to the heart, where it will then get, the blood will get oxygenated and it will be distributed to the rest of the body with these extra nutrients, amino acids and glucose. So the glucose is then taken to the muscles to be used in the process of cellular respiration, which we'll look at as well. And then the fatty acids and glycerol that were absorbed by the lacteals of the villi are now a different story. So let's go a bit down. So the fatty acids and glycerol absorbed by the lacteals of the villi, the lacteals unite to form a larger lymphatic vessel that opens into the thoracic duct. They then enter the left subclavian vein and are transported to the rest of the body via the heart. So just as we had the venules becoming veins and the vein going to the liver, the hepatic portal vein, and into the liver, we now have the a similar thing happening with the lymphatic system. So those lacteals merge and they form part of larger lymphatic vessels leaving the intestine. From there, they go up the lymphatic system and then just behind the heart as well. Then, can you see that area over there? We've got the vein merging with that thoracic duct from the lymphatic system, and that's the left subclavian vein. So these two things merge at that point, and then the fatty acids and the glycerol go into the heart and are then distributed to the rest of the body. I inserted this little table to the left just so you know the difference between glucose, glycogen, glucagon and insulin, just so that you don't confuse, especially the 3 Gs. So glucose is a building block of carbohydrates, glycogen is glucose that is stored, then glucagon is the hormone that increases the amount of glucose in the blood, and insulin is the hormone that decreases the amount of glucose in the blood. I actually think I pasted that in the incorrect area, but now you know. So after the transport of nutrients, and these nutrients are now being distributed throughout the body, how are they used? And this is now the process of assimilation. So cells absorb nutrients to maintain and build cell structures. So all of these nutrients, the glucose, the amino acids specifically with muscles, all of these nutrients are now used to maintain and build cell structures. That is the process of assimilation, short and sweet. Egestion. So egestion is the final part of this long process that we've looked at. So with egestion, the final portion is in the colon. So in the colon, water is absorbed, and undigested and indigestible waste is stored temporarily in the rectum, that area over there. Now, what is undigested and undigestible waste? So there's a lot of food that did not get digested through this whole process, so that just gets egested now. And then undigestible waste. Undigestible waste is food that our bodies cannot digest, and this is generally plant material that is indigestible. And examples of this is lignin, cellulose, and pectin, all found in plants, and they form part of something that our diets need, and that is roughage. So roughage is necessary for effective digestive system. So roughage, another term for it is fiber, there on the right. So why do we need this? It promotes peristalsis, it prevents constipation and hemorrhoids, and then it also softens the feces to facilitate the process of defecation. So after the food has been undigested and indigestible food has been stored for a while, you will then feel the need to go to the toilet and then egest it. So the need to go to the feeling that you receive from going, that you need to go to the bathroom, is because as this undigested and indigestible food gets stored in the rectum, they press against the walls of the rectum, and that triggers some of the neurons, and that sends a message to your brain telling you that it's time to go. But if you don't understand what a neuron is, don't worry, you will learn about that next year. So next is the process of homeostasis, and you'll learn about this next year as well in more detail, but for now, as we go through the different topics, we will look at the different areas of our body that practice this. So homeostasis, once again, if we break up this word to make it a bit easier for us to understand, homeo means similar, coming from the word homo. Then stasis means stable. So basically what this is, it is a constant internal environment that needs to be maintained. So there are different parts in our body that need to remain constant in homeostasis. So the first one is the glucose concentration, your blood sugar levels, then water concentration. We'll look at that when we do the excretory system, carbon dioxide and oxygen concentration, pH, and then temperature. So let's look at the glucose concentration. We spoke about the pancreas previously. So the pancreas is that tongue-like structure that sits behind the stomach. So the pancreas is both an endocrine and an exocrine gland. Exocrine, because if we look at this little duct there, the pancreas releases pancreatic juice through that duct into the intestine for the digestion process. But now the endocrine function of the pancreas is that it secretes two hormones directly into the bloodstream, and that makes it the endocrine gland. So in the pancreas, you've got this little round structure here, and that is known as the islets of Langerhans. Now, in the islets of Langerhans, there are two cells that you need to know of. It's the alpha cells and the beta cells. So what do these cells do? So the beta cells will detect when there's an increase in the blood sugar, and then they will release insulin into the blood. So what that insulin then does is it goes into the blood and it takes that glucose and it converts it into glycogen. Okay, we'll look at that now in more detail. Now the alpha cells will detect when there's a decrease in the blood sugar levels, and then the pancreas or the alpha cells will secrete glucagon into the blood, and then that will cause the glycogen to be converted back to glucose so that the blood sugar levels can rise. So insulin reduces the blood sugar level in two ways. You need to know this. It increases the rate at which glucose is absorbed by the cells, especially the liver and the muscle cells, and then it also stimulates the conversion of glucose into glycogen and fat in the liver and muscles. Okay, so if you've just eaten a meal that is high in carbohydrates, so you've just eaten a packet of Simba chips, or you have just had a slab of chocolate, the beta cells in the pancreas will pick up that the glucose in your blood has just gone up because of digestion and then absorption. That glucose is now in your blood. So what then happens is the beta cells will then release insulin directly into the blood. Insulin goes into the bloodstream and it takes that glucose over here. It takes glucose and it stores it as glycogen in liver and muscles. So now, because the glucose has been changed into glycogen and it's now in the liver and muscles, the blood sugar level starts to decrease until it goes past that constant internal homeostatic level, and then it goes past that and it becomes too low. What happens then is that the alpha cells will pick up that the blood sugar level has now gone below normal, and that will trigger the release of glucagon into the blood. Glucagon then goes into your blood system, and that will then trigger the conversion of glycogen that was stored in your liver and muscles back to glucose. So glucose now goes into your blood and the blood sugar level rises slowly until it gets to normal, and then it will still continue on until it becomes too much. Then what happens again? Insulin is then released, and this process happens the whole day. And this process is known as negative feedback. So there's a detection of a deviation away from the normal result, and this results in a reaction that will counteract what has happened and bring it back to normal. The return to normal is detected and the corrective action is stopped. So in this case, the corrective action is insulin or glucagon going into your blood system. Now, if there's something that goes wrong and insulin or glucagon cannot be released, then it can have some major effects on your health. So the biggest one that everyone should be aware of is diabetes. So what that is, it is high blood glucose levels. And this is a chronic disease, so there's no cure for it. And this is when insulin is absent or insulin doesn't function properly. And this leads to an increased blood glucose level. The symptoms that are associated with diabetes is increased hunger, frequent urination, especially at night, increased thirst. You are always thirsty. You cannot satisfy your thirst. There'll be weight loss, repeated infections, so wounds will heal very slowly. Instead of taking a week to heal, they can take 3-4 weeks. Impaired vision, exhaustion, and dizziness. There are two types of diabetes. There's type 1 and type 2. Not that I'm saying that type 2 is not as dangerous as type 1. They're both equally bad, but type 1 is pretty dangerous. So this is when insulin-producing beta cells are destroyed by the human immune system. So the immune system starts attacking and destroying its own cells, which are these beta cells. And they are very important because they release insulin, which lowers your blood sugar levels. So because they are being destroyed, there's little or no insulin being produced. And the treatment of this is daily insulin injections a special diet, and then regular blood sugar level testing. And this is generally after meals. You'll see that people have these small machines where they prick their fingers, they put a drop of blood on these little strips, they insert it into the machine, and then it gives them a reading. The daily insulin injections, they can also be a bit more serious. They can have a little pump that they have that basically takes over the functions of the pancreas. And I mentioned in class, you even get dogs that are sensitive, that are trained specifically to pick up smells from as humans exhale, to see if there's, for example, if the blood sugar levels are too high, and they'll actually alert the person that they need to check their blood sugar levels, otherwise they can die. Type 2, this is a lifestyle disease, so this is generally because of bad habits. So these people are generally overweight or obese, they are inactive, and sometimes age can even play a role. So type 2 diabetes starts off with insulin resistance. So insulin is produced, but it cannot be used effectively by the body. And you'll see that sometimes if somebody has a certain waistline, that a certain measurement of a waistline, they'll tell you to most likely go get checked to see if you're insulin resistant or even have type 2 diabetes. The treatment and management of this is through a balanced diet, regular exercise, and then oral medication or insulin will also be used in those cases. And that is the end of this whole process of digestion, absorption, assimilation, egestion, and then the negative feedback mechanisms, so how insulin and glucagon work with each other.